Hello, welcome to 13 Universal A-Level CIE Biology. We are on Genetech. We are starting topicals for paper 4 in preparation for your May-June 2024. Please make sure you follow. This is series 1. We're going to do all the topicals and we wish you the best in your exams, okay? So come, let's practice. Let's test what we've learned. Let's check residual knowledge. Bring your writing materials and come with schedule. That's me. Let's roll in this video. Okay, so the first question says therapeutic proteins are used to treat disease. One example of a therapeutic protein is human. Guys, remember, anytime you're studying paper 4, make sure you read the background information critically. So this therapeutic protein is human growth hormone. Yeah. HGH has important roles in growth during childhood and regulation of metabolism in adulthood. Children described as HGH deficient do not produce enough HGF and grow more slowly than other children. People who were HGH deficient deficiency enough, don't have enough HGF and grow more slowly than other children. I've repeated it here. That's repetition. People who were deficient when they were children have a mean adult height, okay, that is, this mean adult height is about yeah, I think it's about 10 point, about 2.5. Yes, 2.5. There's a typo here. Daily injection of HR treatment of HGH treatment for HGH deficient children that can increase growth rate, resulting in an increased adult height. Okay, so look at it here. Let's go on and see. All right, this is 32. All right, now the first question says the growth rate of 3 children was measured. The growth rate of HGH deficient child who did not receive daily HGH, this one did not receive it, okay, was 2.5. Yeah, all right, averagely per year. Then the growth rate of the deficient child who received daily HGH was 10, okay, in the first year after starting treatment. The growth rate of the child who is not deficient was this 5. Calculate the percentage increase in growth rate of the HGH deficient child treated with HGH injections compared to the child who is not treated. So the growth rate, okay, the growth rate, if the child, pause the video and try this video and try to solve the questions. All right, this is a math scale problem. Okay, when the child is not treated, the child grew by 2.5 every year, cm every year. Measure this child's height. Okay, trying to get calculator. All right, so bring out your own calculator. Let's deal with this. So if this child is treated, the child grows from 2.5, all right, to 10. Look at it. Treated 10. Okay, that's 10. When not treated 2.5, so it grew from here to here. So looking at the percentage increase in growth rate. What is the percentage in increase in growth rate, guys? That's what it's asking us. So pause the video and quickly solve it for me. So now increase is 10, right? 10 minus 2.5, right? Yeah, that's the increase, right? So how to solve this? Increase over original. Original is 2.5. Okay, so it was 2.5, right? Okay, please, this is not 2 to 5, it's 2.5. Let me remove it. Okay, 2.5 times 100 over 1. Yeah, this is times 100 over 1. So that would be, that's 100%. That would be 7.5 divided by 2.5. So if you see that's 10 minus 2.5 gives you 7.5 divided by 2.5. Okay, that will give you 3. Okay, times 100. That's 300%, all right, increase. Okay, so showing your working here. All right, so that is it. This is it. Okay, that is calculate the percentage increase in growth rates. So this is the one we calculated was for the child that had it that was not treated. Okay, for the normal child it will be 10 man because normal child grows 5. Can you see it? That's 5 here, right? So this is the growth rate of the child who is not, and that's what a child who is not. So what we calculated was a child who was not, who was deficient. Okay, so but the question is asking for a child who is not, which is 5. So if you are dealing with a child who is not growth deficient, you are going to deal with 5 because the normal growth should be 5. Okay, and then it grows from 5 to 10. But if they ask you to calculate for a child who is growth deficient, you're going to use 2.5. all right, so the question here is asking for a child who is not deficient, so this is it. This is the answer for that. But if you ask for the child who is deficient and grew, that is it for you. Okay, so this is the answer for this question. Okay, all right, but if you ask for a child who is deficient, then you use this. All right, I solved this for you to show you that, but this is it for that. All right, but because it increased from 5 to 10, so 5 is original. Okay, the difference here is 10 minus 5 divided by 5. All right, 2 marks. Let's go to the next question. When this form of treatment started in 1958, HGH could only be obtained from the pituitary gland of people who had died. So they are taking this human growth hormone from somebody who had died to help people who are alive. That's good. So in 1981, a plasmid containing human growth hormone cDNA, that's complementary DNA, was constructed and inserted into Escherichia coli bacteria. This allows recombinant DNA protein to be produced by the bacteria. Okay, it allows it to be produced by the bacteria. So they use this cDNA, right, was constructed and then used to produce human growth hormone. Now, name the type of enzyme that cuts plasmid DNA. So what enzyme cuts plasmid DNA? Try to solve it. Okay, which one cuts plastic DNA? The one that cuts plastic DNA is endonuclease or restriction enzymes. Okay, the one that makes it from human growth hormone mRNA that converts cDNA DNA to mRNA is reverse transcriptase. Okay, so restriction enzyme cuts reverse transcriptase convert cDNA to human. Okay, so identify and explain. So these are two keywords: identify and what explain. So and two properties of plasmid that allow them to be used as vectors of human growth hormone cDNA into cells of Escherichia coli. Pause this video and try to solve this question. Try to solve it. Now I hope you know that plasmids are small. One of their properties is that they are small, therefore they can easily be taken up by cell. Number two, they are circular. Because they are circular, therefore so they are now stable, or they cannot be digested by enzymes because they are circular. And don't forget that they have restriction enzymes. All right, so genes can be inserted. Don't forget they have gene markers. All right, so we can easily identify any genetically modified organism that's taking up our recombinant DNA. Okay, guys, don't forget that also plasmids, they have promoters, so gene can be expressed. They also they can self-replicate. Okay, therefore they can make many copies of that. Look at that, memorize it. Okay, so each of these, because they said identify and explain, so this identification, this small, so explanation, so can enter, self-replicate, and they put, so make many copies. Have restriction sites can be cut by restriction enzymes, so new gene can be started or added. Have markers like antibiotic resistance or fluorescence or filters. Okay, we can easily see it can be recognized. Have promoters, so gene can be expressed. All right, they always ask this question, guys. Let's go to the next one quickly. In 1985, several cases of rare brain disease were discovered in people who had been treated many years previously with HCF obtained from pituitary gland. It was decided from 1985 onwards that only recombinant human growth hormones should be used to treat patients. So the ones made from bacteria should be used to treat patients. Okay. Very importantly, explain advantages of producing human therapeutic proteins such as human growth hormone by recombinant DNA instead of extracting it from dead people. What's the advantage of using bacteria over extracting this from dead people? Pause the video and try it. Okay, taking human growth hormone from a dead person and then inserting genes into bacteria, which advantages does recombinant DNA have over that? Number one, you don't know what killed the person. Okay, so there's less risk of infection. If you're using human from dead people, right, it could be, infected, right? So recombinant DNA technology reduces less infection, right? Less risk of infection. Number two, guys, remember that if you're using DNA, you can have much supply, okay, unlimited supply, okay, because bacteria continues to grow, all right, to make more of this human growth factor in the fermenter. Okay, so purification process is less, so we have less downstream process, so therefore the cost of purification is less. And of course, you know that there are less ethical issues. Don't forget that some people will not be comfortable using what comes from a dead person. Okay, all right, and of course, don't forget that there will be less allergy, okay, or immune response if you use the one from the recombinant DNA. There are lots of them. Look at it here, memorize it. Prevent less lower infection pathogen, cost of purification less, okay, large unlimited supply. Now remember, this also can be asked you using insulin. They ask this question differently. Now look at number 5, potential to engineer or improve. If you're using that from recombinant DNA, you can always improve on it. Okay, 4 marks. Yeah, HG, human growth hormone, is one of the many proteins in the body whose secretion or production is controlled by a person's sleep-wake pattern. The sleep-wake pattern describes when during a 24 hours day a person is asleep and when they are awake. For example, pattern 1, asleep during the night, this is normal, and awake during the day, normal. Now some people are nocturnal, asleep during the day and awake during the night. These guys are nocturnal. Okay, they just switched on. All right, for research, researchers used microarray to identify which genes have their expression changed by a person's sleep-wake pattern. They collected mRNA from a group of people with sleep-wake pattern 1, the same group of people whose sleep-wake pattern 1 was changed to pattern 2, from pattern 1 to pattern 2. All right, for research. Now, a summary of the results is shown in the table. So see sleep-wake pattern, number of genes with increased expression. All right, pattern 1, okay, pattern 2. Now the question says, describe how changing the sleep wake pattern from pattern 1 to pattern 2 affects the number of genes expressed. Okay, so during the day, this is pattern 1, this gene expressed. During the night, okay, this expressed, okay, and all the time this was expressed. So by pattern 2, during the day, this is gene that was expressed. During the night, okay, so you can see that during the day, all right, if you change from pattern 1 to pattern 2, for example, less genes are being expressed. You get, instead of 6 during the day, 661 is now 131. Okay, during the day, less genes are being expressed. But during the night, okay, can you see that there's also a decrease in gene expression? Okay, so few genes are expressed if you change this pattern. Few genes are expressed. All right, now somebody can say more genes are also being expressed because there are genes that can only be expressed, right, when people are sleeping during the day, right? But from this data, because we know that fewer genes expressed have increased, have increased expression. Okay, yeah, so more genes have been expressed overall, okay, because the genes that express only in pattern 2. Okay, they're not expressing. But overall, there's decrease in fewer genes expressed when you switch it. All right, look at this. Explain how eukaryotic genes can be switched on and off, for example, at certain times of the day. Explain. Pause this video and try it. How can it be? How can they be switched on and off? Don't forget that. Yes, they can be switched on and off. Light stimulus can actually, light can, like when light is dictated during the day, right? That light can stimulate the release of signaling molecules, okay, which releases what we call enhancers or silencers. So enhancers allow transcription of certain genes, while silencers or repressors do not allow transcription of certain genes. So that means that some of these enhancers and silencers, they are transcription factors. So the transcription factors, depending on what is released, can either help or prevent RNA polymerase from binding to promoter. Okay, so if what is released in enhancer RNA polymerase can bind to promoter and gene is expressed, but if it's a silencer, RNA polymerase is prevented. So if you mention transcription factors, of course, they're right, they're released. Okay, some of them allow or help RNA polymerase to bind to promoter, some do not. Those are allowed, they're called enhancers. Those don't have silencer DNA sequences. Okay, so the presence of light dictated by the eyes, okay, triggers signaling molecules that cause the release of these enhancer silencer, which are transition factors, and they help or do not help RNA polymerase to bind to promoter. All right, quickly, 2 marks. Explain how bioinformatics can help to identify whether the genes whose expression is changed by removing pattern 1 to pattern 2 are important to health. How do you use bioinformatics? So what is bioinformatics? Pause the video and solve this question. How does bioinformatics help here? Okay, so bioinformatics, how does it help? Bioinformatics, is like that's use of IT in biology. Okay, biological research. So bioinformatics actually is the collection, storage, processing of biological data using computer software. Now, how do we use it? How do you know? So first of all, we compare the genes that are expressed with known genes in the database. So we check, are there these genes that have been expressed, okay, do we have them in database of genes that help in health? Okay, so compare the genes that have been expressed with genes in the database, and then also we now carry out analysis to check whether the genes that have been expressed code for protein that have health benefit. Do you see it? Okay, so see idea of compare known genes. So you compare the genes that have been expressed with known genes in database. All right, so for health, and then of course you do analysis using computer software or algorithm to identify the role of these genes that have been expressed, their impact on health effect. Okay, describe and explain the properties of plasmids that allow them to be used in gene cloning. So why are plasmids? Describe and explain the same reason why they are used as vectors. Okay, plasmids are used. First of all, pause this video and try to solve this question on your own. Pause the video. Okay, remember, describing plasmids are double-stranded. You remember that they're double-stranded? So they're extra DNA material that are double-stranded. So because they're double-stranded, okay, eukaryotic genes can be inserted because eukaryotic DNA is also double-stranded. Okay, yeah, this is plasmid. Now this plasmid are double-stranded, so eukaryotic genes can be inserted. Now this plasmid also, you can see that they are small. Because they are small, they can easily be taken up by bacteria. Now you can see that they're also circular. Circular means that they are stable and they cannot easily be digested by enzymes of the host cell. Okay, don't forget that they have restriction enzymes or sites where they can be cut, so genes can be inserted. Don't forget they have gene markers, like fluorescent genes, genes for antibiotic resistance, so they can be identified. Fluorescent genes like GFP gene, GFP gene that code for this protein that when you shine UV light on the organism, it emits green light, or GUC, glucuronidase All right. Alpha glucuronidase. Beta is beta. Okay. This particular enzyme converts colorless substrate to color product. Okay. So any bacteria that have this, all right, gene in a plasmid, if it has a recombinant DNA, we can use it to identify it. Okay. Or antibiotic resistant gene. So it has it. Of course, some of these plasmid have promoters, so they can, any gene inserted in them can be expressed. Okay, they can self-replicate, can make many copies. Look at it, guys. Memorize. They was asking this question. Double-stranded, one mark, have precision site target. Okay, all allow gene for cloning to be inserted. Small plasmid can enter. So these are one mark circular. So stable, contain markers can be identified, replicate fast. So get many copies, many, right? So look at, explain the role of recession endonuclease, ligases, reverse transcriptase in genetic engineering. So pause the video and endonuclease and try this question. Pause the video. Endonuclease cut DNA. They used to cut DNA. They're called molecular scissors. So they used to cut DNA at the restriction site, palindromic site, okay, to produce sticky ends. So if they are cut in a staggered fashion, sticky ends are formed. Sticky ends are short tails, okay, at the end of the fragment that are complementary to another fragment of DNA cut by the synthesis enzyme. So that's nuclear. But if they are cut in a straight fashion using precision endonuclease, they form blunt end. So ligases are used to join two fragments of DNA to form sugar phosphate backbone. They seal up sugar phosphate backbone and the phosphoester bond to perform recombinant DNA. Okay, reverse transcriptase converts cDNA to what? Converts mRNA, sorry, to cDNA. All right, look at it, guys. Yeah, see, cut DNA for this adhesion site, palindromic. Okay, sticky. Join plasmid, gene to plasmid. Okay, ligase join the gene to plasmid. Gene is human gene, this is bacteria. So, and that will give us recombinant DNA. Okay, so it forms sugar phosphate backbone, seals it for us, phosphoester bond or covalent bond. It makes cDNA from mRNA. So these questions are the same. 8 marks. In 1984, geneticist Alec Jeffreys invented a DNA testing technique known as DNA profiling that produces a DNA banding pattern on a gel. The DNA banding pattern profile is unique to each individual. DNA profiling can be used in police forensic, work to catch criminals. Since 1987, police in many countries have collected and stored DNA from crime scenes to create DNA profiles. Awesome. The biology has real life application, guys, which they try to match with DNA profiles of criminal suspects. Okay, DNA at a crime scene may be obtained from hairs, traces of blood, semen, saliva. Explain why PCR may be needed before DNA from a crime scene can be profiled. Now, before you profile it, pause the video. Why do we need PCR? What is PCR? What is it used for? Okay, what is PCR? Pause the video and try this question. Why should PCR be needed? Okay, PCR is called polymerase chain reactions, used to amplify DNA molecule in a short time to make multiple copies of a section of DNA in a short time. So why do we need it? We need it because the DNA from crime scene they're usually in small quantity. So you can't do enough profiling with small quantity of DNA. So you have to amplify or multiply the copies of DNA using PCR. Okay. See, crime scene DNA may be present small, so amplified. 2 marks. In 1984, the geneticist Alec Jeffreys invented a DNA testing technique known as DNA profiling that produces the DNA. I think we've read this. Okay, yeah, we read this. Okay, so let's look at the second question. Explain why electrophoresis produces a DNA banding pattern on a gel. Why does it produce a banding pattern? Pause the video. Why does it produce a banding pattern? Give us a reason. This is an electrophoretic electrophoregram. Okay, and then wells are cut here. Remember, DNA samples are loaded, DNA samples are extracted, they're denatured, amplified, and then they are mixed with fluorescent tide and loaded into this well. Wells are cut into agar gel, and then of course this is positive electrode. All right, these DNA samples are going to move from negative is cathode, positive is anode. So why are the band patterns? See, at the end, if you apply electricity here, you produce different band patterns. Okay, band patterns. Yeah, why do we form? What is the principle behind these band patterns that are unique? These band patterns are unique to human beings. So human beings have different DNA band patterns. We call genetic fingerprinting. Okay, this is for this guy. Now for this guy, sorry, this guy has his own. Okay, why? Have you found the answer? This is because DNA is negatively charged. I hope you know that the charge of DNA is negative because of the presence of phosphate group. Phosphate group is negatively charged, right? So, and again, because it's negatively charged, so the DNA fragment always move, okay? It moves from the negative electrode, which is cathode, to the anode, all right? Yeah. So they move from here to there because they're negative charge, and of course the short fragment move faster. So this one is the shortest, this is the longest for this guy, so we can match it. This matches with this, so this guy was in the crime scene. Okay, so the reason is because DNA is negative charge and then it can move. So look at it, DNA moves to positive, and okay, smaller light fragment move faster when electricity is passed. GED Match is described as an open data personal genomics website. It can be used by people who want to upload their DNA data to trace their ancestors and other relatives, like some of the blacks lost, everywhere in the world, black Americans everywhere. They are looking for their ancestors to connect, okay, within these countries and outside. So this can help them, but the police is using it to solve problem. Let's see what they have done. In 2018, police in the USA solved a large number of serious crimes. Some of these crimes had been unsolved for over 30 years. That means these criminals have been walking free, causing more havoc, and the innocent people are paying for it. All right. The police used GED match to profile DNA taken from crime scenes and to look for matching DNA profiles. In many cases, the police found partial matches to relatives of criminals. This allowed the criminals to be identified and then charged on the basis of complete DNA profile match. So see, biology is solving real life solution. So just why the police strategy of comparing crime scene DNA with GED match, why was it so successful? Why was it successful? All right, pause the video. When they use the word suggest, think outside the box. Why was it successful? Use imagination. What made it successful? So suggest is a command word that makes you think outside the box. Come out of the box. Okay, why did it work? We have GED match on the internet and they will have police database. Wow, okay, why did it work? Okay, so I think that why it worked because this GED match, right, already has the DNA profile of innocent people. People who have nothing to hide. They put it there because if you have something to hide, you will not put it there. So most people there are innocent. Okay, and then the police database has people who are suspected or convicted of crime. See, so the GED match has is larger. The database is larger than that of police, and of course, it could be used as a lead. So people who are suspected can be matched with people whose DNA is in GD match in GD. Okay, DNA can match them, right? So they could lead us. So those of them that are suspected of crime, right, that has partial match, right, can be used to trace those who are criminals. Okay, so GD match database is larger than police. So a lot of people are there. Then police data only covers people convicted. See, so GD match has data for innocent people and then who don't hide anything. So provide leads to people who are hiding. Okay, all right, too much. Okay, the same question here. Let's see the other follows. I explain why GD match is an example of bioinformatics. Okay, pause the video and try it. Why is it? Okay, bioinformatics. Okay, the reason why it is used as bioinformatics is because it is a database. So it stores DNA, okay, nucleotide bases of people, or stores the genome of people. So it's a database, and bioinformatics deals with that database. Number two, it can be used for analysis or processing using um, algorithm or computer software. Okay, yeah. So these are things that are found in bioinformatics. You can talk about store of contains large biological data for analysis and computer software. So that's why GED Match is an example of bioinformatics, because it has the features of bioinformatics. The first successful conviction resulting from the use of GED Match by the police was widely reported. Some journalists and broadcasters thought that the GED Match website should not have been used by the police in this way. So people got emotional. So why should we use police is exploiting people? The police are exploiting people. People are putting up, their genetic information on the internet, and the reason why they are doing that is to find their ancestors. Now police is taking advantage of that. That's not fair. That's not the major reasons. Okay, you are a bioscientist, and now we need your opinion. So in the days following the news, the number of citizens choosing to upload their DNA data to GD Match increased. Why the broadcasters are protesting? More people started. Why did more people? What motivated more people to upload their DNA data? So can you see it increased from 1,500 to 5,000? What motivated people to do that? People are living in fear, guys. Now we have solution. Okay, comment on the social and ethical issues raised by the first successful conviction. So what are the issues here? Right? What is that? When they say social and ethical issues, we're talking about what advantages, problems, what advantages does this, right? Use of GED match. What advantage? Number one is that many crimes were solved, criminals were caught. Okay, that's what people say uploading it. And then, because criminals were caught, it created a safe community for everybody. So people want more criminals to be caught. Okay, yeah, and of course, finally, we now see that DNA has brought brought order. Okay, it has helped the judiciary system. These are ethical issues. However, another problem is that people believe that sharing one's DNA data, right, also reveals the DNA data of your relative, and you should seek their consent. Okay, that's it. So help police catch criminals, make sense safe. Okay, so this has to do with whenever you hear ethical issues, social advantages, disadvantages, and problems, then a breakthrough self justice, law and order, sharing. Yeah, this is it. Okay, okay. Now, yeah, adenosine deaminase ADA deficiencies and immune system disorder caused by recessive autosomal mutation. Severe combined immunodeficiency caused by a lack of ADA is called ADASCID. So this ADASCID, adenosine deaminase, all right, helps to deaminate adenosine and deoxyadenosine because they are toxic to T lymphocytes. So if the enzyme doesn't work, these adenosine and then deoxyadenosine will destroy T lymphocyte. And T lymphocyte, once they destroy, there will be weakened immunity, right? And these people suffer from severe combined immunodeficiency. Any small infection can affect them. Genetic engineering is used to make a recombinant human protein to treat people with ADA. So we're solving problems. Scientists are solving problems. Okay. So the question is, so let's see the question, guys. Outline the principles of genetic engineering. Anytime they ask you to outline the principles of genetic engineering, guys, they're just asking you the procedure. That's it. They're asking you for the procedure. So what is the procedure for recombinant or genetic engineering? What is the procedure, guys? What is the procedure? That's what they're asking you to do. Pause the video and try it. Now, I think the procedure, of course, you cut out, obtain the gene from another organism, okay, and then you cut plasmid. Also, so you cut out gene using restriction enzymes, and then cut bacterial plasmid with the same gene to form complementary sticky ends. Then insert the human gene into the bacterial plasmid to form recombinant DNA, and then transfer the recombinant DNA to the host organism. Okay, of course, when you transfer it, what happens? Then the gene must be expressed there, so the gene can be expressed, and the new organism acquires a new trait. Can you see? Add DNA to give a new unnamed characteristic protein. Okay, so add DNA there. Of course. Okay, so you see it. So outline the principle. Okay, this is just the procedure. So you're adding DNA to give a new characteristics, all right, from another organism. So where do you get this DNA? Because DNA, all right, contains genetic information for proteins that control traits. If you need trait, get a gene for that trait. So first of all, get the gene using the restriction enzyme to obtain the gene section, cut plasmid with the same, okay, nucleus, endonuclease, and then combine the gene with the plasmid or vector and introduce it into the bacterium, clone it. It clones, multiplies inside the GM, and then the gene is being expressed. It's just anytime you hear principles of genetic engineering, is the procedure, the step by step. In 2016, gene therapy to cure ADS-KID was approved in Europe. Okay, that's a good news. The gene therapy involves three main steps. Blood hematopoietic stem cells are taken from the bone marrow of the person with ADS-KID. The functional gene and its promoter are inserted into the blood stem cell. So they put the gene into the blood cells, okay, with the promoter, maybe using viruses as vectors. A single infusion of the gene-corrected cells, that's gene-corrected stem cell, is given to the patient. Explain why just a single infusion of gene-corrected stem cell is enough to cure disease. Why is that one single infusion? What is it about stem cell that one single infusion does the job? What is stem cell? So you have to connect it. Okay? Pause the video and try it. Pause the video and try it, okay? So remember that stem cells divide, okay, by mitosis. They proliferate to make more cells. So if one stem cell has it, it will divide and then pass on the gene to all the new cells being made. So stem cells can make a lot of cells in a short time by mitosis, because it divides continuously by mitosis and then passes on this corrected gene. Okay, that's it. Stem cells divide, multiply, make new cells, and this gene is inherited or passed on. That's all. Okay, the same. Explain why a promoter has to be transferred. Now, what is the purpose of a promoter? Okay, pause the video. Anytime they ask you a question on promoter, pause the video and try it. Number one, promoter helps gene. It helps transcription, okay, of genes. For genes to be expressed, that means it allows RNA polymerase to bind to the DNA and then switch on the gene, cause the gene to be expressed, so that the protein can be made, mRNA of the gene can be made to switch on, okay, at right time. They put at right time is sufficient to allow bind RNA polymerase. Memorize it. They ask questions a lot from promoters. Okay. A modified retrovirus is used to insert the new gene into DNA. State 2 ethical consideration using a retrovirus. Remember what I said? Ethical issues, the advantage, disadvantage, the problem it creates. Does the virus create problem? Pause the video and try it. Yes. If the virus can disrupt normal genes, okay? It can insert the gene. Now the virus is acting as a vector here. It can insert the gene into the wrong position or disrupt another gene. Okay. Now if the gene it disrupts is a regulatory gene, it can cause cancer. Right? And viruses are parasitic. They can cause infectious disease. Right? They can also trigger immune response. You get? So this is it, guys. All right. Okay. Must not insert in wrong place. Could almost cause cancer. All right. So they're always asking this question. Memorize it. Thank you, guys. Please make sure you share the video. Let your friends who are preparing for this exam have this video. Okay? And tell us what you want. We're here to serve you. This is Shedro. Don't forget that these videos, the questions come from CIE past papers and masking. So have a lovely time. See you next episode. Okay? Next series. Bye.